So thank you for the introduction, Patrick, and no pressure after that introduction. I'm going to talk to you about chikungunya. I'm going to talk to you a lot about the virus and also about the vectors because this is a very different animal than West Nile virus, and so you need to really switch gears on how you're going to deal with this and understanding this virus. Before I get started, I do need to make a statement that the what I am presenting here today is the opinion of the author and not necessarily the representing the policy of the Centers of Disease Control. The reason I have to actually say it out loud instead of letting you read the little bitty fine print on my title slide is the camera in the back of the room. So for both of my presentations today, just in case I forget this afternoon to say that, this is my opinion. So as everybody in the room should be aware that we are just a plane ride away from exotic diseases around the world. This map represents just a single time point of airplanes in the air. There's a web page you can go to and you can actually see a movie of the airplanes as they are moving through the sky, and it's just this, map with all of these lines all over it because there are thousands of aircraft in the air moving between countries, moving potentially ill people to new places where a disease may not be endemic. And a case in point, in 2013, we had 772 imported cases of dengue where people were traveling or living in a dengue endemic area, and they hopped on an airplane, they came to the United States, they became ill, they sought medical help here, and were diagnosed with dengue that they had picked up somewhere else and brought it back. So I want to talk a little bit about arboviral zoonosis and transmission cycles because you guys have probably seen this cycle before where for most of the arboviral diseases, and arboviral or arbovirus does not have anything to do with trees. It is a abbreviation for arthropod-borne viruses. So there needs to be an arthropod within the transmission cycle of the virus. Usually there's a vertebrate host for most of the viruses, the endemic viruses in the U.S. There is a vertebrate host. It's not necessarily always a bird. Sometimes it is a small mammal. But you have the virus which is picked up by a vector from an infected host, and then it's transmitted to another host. By the same way that infected mosquito bites a uninfected host. Where we run into problems with that is when it starts spilling over into humans or incidental hosts. And most of the arboviruses in the U.S., humans are a dead-end host. We're not important in the transmission cycle, so that cycle goes on out in nature without us all the time. And then also humans are considered a dead-end host because they don't have enough of the virus circulating in their blood for a mosquito that bites them to become infected with the virus and transmit it to something else. So that's why we are a incidental host or a dead-end host. And where we really get into problems is when the abundance of the mosquitoes, particularly infected mosquitoes, becomes great enough that you get a lot of people becoming infected. So I'm going to talk about chikungunya. This representation up here is actually a pretty simplistic representation of what goes on in the environment in these life cycles. They're actually very, very complex. And a large part of that complexity comes from what's called the intrinsic incubation period. And this is a period that when a mosquito takes a blood meal from this first infected human, which has viremia or virus circulating in the blood, that mosquito takes that blood meal, and there's a period of time where that virus has to multiply in the mosquito and get from that gut to the salivary glands, so that when that mosquito takes a second blood meal and feeds on a second human, they can actually transmit the virus. And then there is an intrinsic incubation period, which is the time from when that person is bitten to the time that they actually have the virus replicate in their blood, in their bodies enough that it's circulating and that they can then be infective to the next mosquito that comes along and bites them. And so there's actually quite a complex just interaction between the vector and the humans themselves. And then just think, if you have to have a bird in that, how much more complicated that cycle might be, which is what happens with West Nile virus. But in addition to what's going on between the vector and the host, you also have a whole host of other things that affect the cycle, particularly the arboviruses that we're used to dealing with, like West Nile virus, where we have weather and climate. You have to have that extrinsic incubation period is driven by development of the mosquito. It's driven by temperature, so that period can be longer or shorter depending on how cool it is. There's predators, pathogens, there's everything that goes into what the vertebrate host needs to survive, in abundance of the vertebrate host. Also, there's everything that goes into the abundance of the mosquitoes as well. So you're probably used to thinking about vector indexes, which are a combination of the abundance of the mosquitoes and the numbers of mosquitoes that are infected in the environment. And for almost all of our arboviruses, really the only place that we can make an impact on them is controlling the vector, because we don't necessarily have vaccines or treatments when people get sick. So there are two vulnerable spots in our virus transmission cycles in the U.S. And again, there are vaccines for animals, for horses, to keep them from getting sick. But really, this controlling the vector is our only option. And that's no different with chikungunya. However, this whole life cycle I'm talking about, this is our native viruses. This is West Nile virus. This is Eastern equine encephalitis, St. Louis encephalitis. All of these viruses have this animal component in the environment. Chikungunya is different, and I'll get into how it's different. The general life cycle of the mosquito is very important to understand. This is a representation of the culex. You guys should be familiar with this, with West Nile virus. They lay their eggs in rafts on the water, and then they go through the 4 larval end stages to the pupal stage. They emerge, they go out and they get a blood meal, and they start that whole cycle again. Aedes aegypti is different in that it does not lay its eggs in rafts. It lays them individually. So where you find the eggs and where you find the larvae is going to be a little different than where you're going to find culex predominantly. And then also what they feed on is very important as well. So the culex for West Nile virus, they actually like feeding on birds, probably more than feeding on humans, whereas the Aedes prefer to feed on humans. And then you guys have probably also seen this for West Nile virus or any of our arboviruses, where What you see clinically in humans is actually the tip of the iceberg as far as the numbers of people who are actually infected by West Nile virus. So most of the people who are infected by West Nile virus, about 80% of them, don't show symptoms, or they just feel a little bad for a day or two and never go see the doctor because they just think it's a flu or a cold, and then they get better. Whereas about 20% of them actually get sick enough to seek medical attention. And then a small portion of that get the really severe forms of the disease. So we're going to talk about a whole new animal here. We're going to start talking about chikungunya after that brief review of arboviruses in the U.S. So chikungunya is a flavivirus. It's in the family Flaviviridae. It's an alpha virus. It is associated with urban, suburban, and rural habitats. So this is found not just in the cities, but is also found, at least in its native home range, in rural areas. There is no vaccine for it. The primary host, this is where it starts getting different from West Nile virus. The primary host is humans, okay? So this virus can circulate. Humans mount a high enough viremia that mosquitoes feeding on the humans can become infected and transmit it to other humans. There are secondary hosts to this, particularly that we know of in Africa and in the Old World. Non-human primates, rodents, bats, and other small animals have also been implicated as being important in the life cycle. And I'll talk a little bit about that because, quite frankly, when West Nile virus came, we didn't quite know what to expect. We thought we knew what it was all about from experience in Africa and Europe. This is the same way with chikungunya. Now that it's in totally new virgin habitat, we don't know how important secondary hosts are going to be. The vectors, Aedes aegypti, Aedes albopictus, these other species that are listed up here are African species, and they are jungle species. So here in the U.S., you're not going to be seeing Aedes crucifer or Aedes africanus. But we don't know what other local Aedes might be important in maintaining this virus. So the chikungunya transmission cycle is actually composed of two different cycles. So this is where it really starts to get different from West Nile virus. It actually acts more like yellow fever than it does like West Nile virus. So you have what's called a sylvatic cycle, which happens out in the jungles in Africa, where you have these non-human primates that are being bitten by these forest mosquitoes, and they are able to maintain chikungunya in a sylvatic habitat. And then you get spillover that when humans go into the forest, they get bitten, they bring the virus back to their town or to the city that they live in. And then you get this urban cycle that occurs. And this is where it really gets fun, because the outbreaks that we see of chikungunya virus are these huge, very intense, very hot outbreaks. So this cycle, and again, this cycle can maintain in urban environments without any of these other mosquitoes and without the monkeys being involved in it. So just as a recap, when West Nile came to the U.S., when it got here, we found over 67 species with the virus in it when we started looking for West Nile virus. What this means for chikungunya is as far as additional vectors that might be native to the U.S., like Aedes vexans or some of the other Aedes. We don't know how that's going to end up playing out. until the virus actually gets here and if it really starts establishing. There were over 300 species of birds that West Nile virus had been identified in. Remember, we don't have that wildlife component of the life cycle for chikungunya. So it really doesn't matter what the wildlife is doing because it's the human mosquito-human that's going to maintain the outbreaks when it's here in the U.S. However, with that said, in the old world, there is that wildlife component within the monkeys, and we don't know how that's going to play out here in the U.S. Or in the Americas for that matter. We don't know if the monkeys in South America, Central America, are going to be competent hosts that can maintain a sylvatic cycle in the Americas. We really don't know any of that. So, and we also don't know if some of our native rodents or bats or might become important host of the virus here in the U.S. because the virus did not evolve in this hemisphere. So all of this is a totally unwritten work right now. There are currently 2 lineages of chikungunya circulating in Africa and in Asia. Relatively recently, I say relatively because it's been some years, actually about 8 years, 7, 8 years. There was a mutation in the African strain which made Aedes albopictus a super spreader of the virus. So the primary vector really was Aedes aegypti for both of the strains, and then after that mutation occurred, Aedes albopictus became much more efficient at spreading the virus. And then the incubation period, time from infectious to illness in people, can be 2 to 12 days. It's usually 3 to 7 days, but that viremia occurs during part of that incubation period. So you can actually be sick with chikungunya and bitten by a mosquito and infect that mosquito before you are showing symptoms of chikungunya. But that period is only a couple of days, and then you're sick, and then at that point, when people are sick and they know they have chikungunya, the recommendation is to keep mosquitoes away from them so that they cannot infect the next group of mosquitoes to transmit chick. It's characterized by an acute onset of fever and severe polyarthralgia. I hope I said that right. So that's where the word chikungunya comes, or the meaning of the word chikungunya is that which bends. So people who get this disease, they have these severe arthritis-like symptoms, and they manifest mostly in the small bones, so your hands and your feet become incredibly painful and swollen to the point that people who have this disease literally can't get out of bed to go to the bathroom. They're in so much pain for moving. So it can be a very, very, very painful disease, although there's very little mortality associated with this disease. The arthritic type symptoms can actually linger for a long time. So the acute phase of the disease, where you have the high fever and you're really, really feeling sick, will resolve itself in about a week. But that lingering pain in the joints can last for months and years beyond the acute illness. And what happens in large outbreaks is that a large proportion of the population tends to get infected with this disease. So it really burns through a community with a lot of people getting sick. And unlike West Nile virus, most of the people who become infected with this disease are going to be sick enough to seek medical help. So if you think about that iceberg that we have with West Nile virus and our native encephalitis viruses, arboviruses, flip that iceberg upside down, because you're going to see anywhere from 70 to almost 95, 100% of the people who are infected with this virus showing clinical signs of it. And for that reason, actually, most of our arboviruses, humans aren't very good sentinels. With chikungunya, humans really are the sentinel, because they're getting sick before you're likely to start picking it up in mosquito pools. And you're going to see those people, and in a pretty short period of time, because it only takes a day or two for them to get sick once they're bitten by that infected mosquito. So really only a small proportion of the people who are infected are going to be in that subclinical class. And then attack rates of the general population are going to be quite high. How that plays out in the U.S., we don't necessarily know, because this is a tropical disease and our housing is not tropical housing, and our mosquitoes are not living inside of the houses with us for the most part. So chikungunya, the greatest risk of getting chikungunya established is in areas where dengue fever is established. And that's because the vectors are the same for both viruses. It's the Aedes aegypti. Both viruses are a human primary host virus without that wildlife component in it. And so the conditions to support epidemics are going to be the same for chikungunya and dengue, the conditions that are required. So this is a map of the global dengue risk that came out in the New England Journal of Medicine. And so where you have the orange are areas where people are at the greatest risk of getting dengue fever. And so we postulate, because these two viruses have such a similar cycle, that these are, and share the same vector, that these are going to be the highest areas of concern for getting chikungunya. And in the Americas, I want you to kind of pay attention to where the dark red is and where the blue is, because when we start looking at where chikungunya is, now that it is in the Americas, I want you to compare those two. So because the two vectors, Aedes aegypti, Aedes albopictus, are in this hemisphere, this is the rough distribution of albopictus and aegypti. It doesn't mean that aegypti is everywhere in this area, but this is the native range of Aedes aegypti. Aedes aegypti is starting to make a comeback. We are finding it in more and more places where it had once been, and then Aedes albopictus came in, and we couldn't find the aegypti anymore. Well, now we're starting to see more and more aegypti re-emerging in its historic range. But also remember that there was a shift in vector competence with the African strain, making Aedes albopictus a super spreader. Luckily, what we have in this hemisphere is the Asian strain, but vector competence studies that Ann Powers' group at CDC has done shows that our Aedes albopictus here in the United States is just as good at vectoring, at harboring the virus and growing the virus as our Aedes aegypti local populations. So just a little bit of history. 2007 is really when chikungunya started moving out of Africa with local transmission in Italy. This is really the home range of chikungunya, It also started marching out of Africa across the island hopping across the Indian Ocean into parts of Asia where it had never been. And then in actually late 2013, the first cases in Saint Martinique were diagnosed. So in October of 2013, it had hopped over to the islands in the Caribbean. This information is from January 2014. So really only these couple islands in the Caribbean at that point had cases. As of February 24th of this year, it has moved out of this initial introduction throughout most of the Americas. In fact, at this point, it's actually easier to list the countries that it's not in versus the countries that it is in, because it's in 40-something countries in the New World. And the numbers in parentheses, these are imported cases that have been detected. So even though they don't have local transmission going on in these countries, they do have people bringing it into these countries. And so it is a matter of time, probably, until these countries have local transmission, with maybe the exception of Argentina and Canada, because I don't know how many Aedes aegypti or albopictus are in the extreme north and south of the continents, because again, this is a tropical disease, not a temperate disease. And that map, you know, looks really scary because it's all that dark green with countries that have chikungunya. But if you look at a little finer resolution of the places within those countries where local transmission is occurring, you see that it's not uniform. So in the country of Brazil, really it's only parts of Brazil where chikungunya is. In the United States, really it's only in Florida where local transmission has occurred to date. But I want you to remember, looking at the dengue risk maps, what it looks like compared to what we're seeing with the chikungunya. And it really is playing out that the areas at highest risk for dengue is where chikungunya is being detected. And so I think we kind of made the right call. I do want you to look at where Texas is on this map, though, because Texas is in a high-risk area for potential dengue transmission and therefore for chikungunya transmission. So if you want to look a little more in depth at chikungunya, CDC has on their website, if you go to www.cdc.gov and just search chikungunya, before it even made it to the Americas, we had worked with our partners throughout the region and developed some preparedness and response guidelines. It's a pretty handy reference point for people to use. And then because there's a lack of effective interventions, there really is no vaccine. There are no clinical trials in progress for a vaccine. There's not going to be a vaccine anytime soon. Because of that, we're really left with vector control. And currently, the way vector control is implemented in countries with dengue, where dengue is endemic, we're really not making much headway. And so with chikungunya, it's just another virus, but our current vector control tools are not, we don't know how to use them effectively at this point. And then most of our information on the vector control tools that are out there are from dengue endemic areas, and I would say that in the U.S., we're not necessarily in the same situation as we would be in southern Mexico or Saint Martinique or Puerto Rico for that matter. So we have some needs that really we need to meet with this virus. Personal protection behaviors, You know, we usually throw that out as a front line. Well, just don't get bitten by a mosquito, and you won't get this disease. But I would challenge anybody in this room to wear repellents 24/7, 365 days a year to prevent themselves from being bitten by a mosquito. I think that message, while it's great politically that, oh, we don't have to, you know, do a lot of spraying or use all these chemicals, people just need to wear repellent, is pretty unrealistic. And so as public health educators, yes, you need to tell people if they're going to be out when the peak biting period of the vector is occurring, these people can be bitten anytime during the day or night by these vectors. And so giving people this false sense that if they're wearing repellents, they're not going to get the disease is really a disingenuous message. I think that we need to make a more concerted effort at explaining that to people. But some of the needs that are coming out of chikungunya, again, is improved vector surveillance, control, and tools. I'll talk a little bit about it, but the speakers coming behind me are going to have a lot more to say about vector control and vector surveillance. And then evaluation models for spread potential. I think we have some pretty good models out there with our dengue models. And they seem to be coming along and proving us right that where we're seeing the cases of local transmission are in the high-risk dengue areas. But we also need a little bit of improved awareness about other viruses because last year I talked about chikungunya, but I also mentioned this virus down here called Zika virus. It too is moving, and Zika virus has moved over the last year across the Pacific and is now as far east as Easter Island, so it's just like the last islands before it hits South America, so stay tuned for maybe another virus next year. So I talked about the symptoms already. Again, I want to reiterate, though, that patients with symptoms, 12 to almost 20%, and depending on the study you look at, have symptoms up to 3 years after they become infected with this virus. So even though it's not a killer, it's going to be causing problems for people for a long time after they get infected. And even though up to 47% of people had symptoms at least 2 years after infection. So again, there's this acute phase where you're really, really sick, but this virus has a substantial chronic phase associated with it. And just to do a little bit of comparison between chikungunya and dengue, because it's often very, very difficult for a doctor to distinguish between the two viruses because they have so many symptoms in common, but there are some differentiating symptoms like this arthralgia, lymphopenia, some of these but a lot of the other symptoms between the two are shared. And the reason why it's so important is that clinically, if you have dengue, you don't want anybody to give you aspirin or ibuprofen or any of these pain medicines that are going to make you feel better. Whereas with chikungunya, because you don't have a hemorrhagic form of it, it's okay to have these other painkillers, but not ideal. And then the other big difference is with dengue shock, what a lot of people, when you go into shock, it's because your blood volume is low, and so people, doctors will give you IV and fluids to bring the volume of your circulatory system up. Whereas with dengue, if you do that, you actually make the people worse because their blood volume is low because they're leaking, and you start adding more fluid in there, and you just cause more problems. Whereas chikungunya, if somebody's going into shock, that's, again, not an issue. So the way that they're managed clinically is different, so it's very important to know the difference between the two. So finishing up on just the biology of the virus, in the Americas, as of February 6th, there were over 1 million cases, well over 1 million suspected cases in the Americas, with 181 deaths. So again, it's not a very deadly disease, but there's a lot of it, unlike West Nile virus when it first came to the hemisphere. In the U.S., there were 11 locally acquired cases in Florida. These are provisional numbers because sometimes we get late reports, although we're about to cut off the numbers for 2014 and make them official. For almost 4,500 locally acquired cases in Puerto Rico and the Virgin Islands. So it's in the U.S. territories, and a lot of imported cases in the U.S., across the U.S. So just about every state has had a imported case reported from it in 2014. And I want to leave you with a sobering thought, is that at the end of February of this year, we already have imported cases being reported to us. So there's not a winter going away of this virus, at least as far as it moving around and being imported into the U.S. And the numbers here, Florida's already reported 12 cases, New York 9, Maryland 4, Texas, Missouri, and New Jersey have each had 3 cases. So there's been 3 imported cases here in Texas already this year. And then in places where transmission really is occurring, in Puerto Rico, and this should be an I, Virgin Islands, it's locally acquired cases are already circulating in the environment. So let's talk a little bit about these vectors, the vector biology part of my talk. This is Aedes aegypti. She has that nice lyre-shaped marking on her thorax versus Aedes albopictus, which has the single stripe. The biologies of these 2 mosquitoes are very similar. However, Aedes aegypti really is a domestic mosquito. You rarely find it breeding in natural breeding sites, such as tree holes. It likes to be down in the trash and the trays under the flower pots. It likes to be really in man-made containers, whereas Aedes albopictus will exploit a lot broader habitats as far as laying their eggs and their larvae. So you can clean up all the trash in a neighborhood with albopictus and have a much more difficult time bringing the adult numbers down because they are up in the trees. Also, Aedes aegypti wants to bite you. They don't want to bite the dog. They don't want to bite the squirrel. Whereas Aedes albopictus, why it does better in the trees in suburban areas is because it will utilize those natural cavities holding water, but it'll also utilize the animals around it as well. So it'll bite the first thing it comes across that has blood in it that's a mammal. Approximate distribution of albopictus at this point in the U.S. and Aedes aegypti. This is changing. Every day they're finding more aegypti in more places, and albopictus is continuing to spread north and west as well. So there's a lot of ways that we survey for mosquitoes, and Tom is going to talk about surveillance more, so I'll skip a lot of this. Larval surveillance is you're going to be looking in places like this more than in permanent water, because these are not permanent water breeding mosquitoes, and they do prefer to be in storm drains, in water collection containers in yards. Some of these other things up here are not efficient for surveying for albopictus or Egypti. So what is vector surveillance all about? It's about counting mosquitoes, how many there are, where they are, when they are, and how reliably you can do that is going to dictate the information you have as far as risk for chikungunya virus in a particular neighborhood. Aedes, albopictus, and Egypti are very difficult to catch in traps. They don't respond well to lures, so they're really hard to survey for. They are very highly visual, whereas Culex quincunfasciatus uses the sense of smell, and so you can fool it with CO2 and other lures to enter traps. Albopictus is a very visual hunting mosquito, and not so much using its sense of smell or detection of chemical. And it's incredibly difficult to evaluate impact of control measures on these mosquitoes. So the two most common traps, these are used in West Nile virus. Everybody in this room is probably very familiar with the CDC gravid trap and the CDC CO2 baited light traps. These are very, very efficient traps when it comes to finding West Nile virus vectors or vectors of our other arboviruses, but they do not work for Aedes aegypti. You will not find Aedes aegypti going to a CDC gravid trap. You really need to know your mosquitoes and the differences between them. So these traps work well for Culex pipiens, Culex quincunfasciatus, which are night biters. Aedes aegypti is primarily, I say a day biter. It actually has 2 peaks in its biting period at dusk and dawn, but it will bite throughout the day. It'll bite at high noon. If you go into a shaded area with Aedes aegypti or Aedes albopictus, they will bite you. And they will also bite at night. So that's not a peak biting period for them, but again, if you have them on your deck breeding in the plants on your nice pretty deck, and you walk out there in the evening, they will take advantage of that and bite you at night. Intensive larval control programs have been shown to work, but they're really, as far as cleanup programs, not very sustainable. And again, it doesn't enter gravid traps. It's a container mosquito versus a permanent water mosquito for the pipiens. And the 2 ways that we really survey for it are with these little black jars or OV traps, and then what's called the B&G trap. And Tom, you're going to talk more about the traps, so I can skip over talking. Are you? Yes, you. I'm sorry. Okay. Because I can talk more about them. But I don't want to be too redundant for the speakers coming behind me. One of the key things about the behavior of both of these species is that they do what's called skip oviposition. So Culex, they lay their eggs in that raft. All of their eggs for that blood meal are in one raft in one site. The Aedes will do skip oviposition. So this mosquito, she lays her eggs singly instead of in a raft. She's going to come along. She'll lay 1 or 2 eggs in the first container she finds. She'll fly on and lay a couple eggs in the next container she finds. So she's going to spread, that one female is going to spread her eggs throughout the environment, throughout multiple larval sites. And another thing about the 80s, which makes doing egg counts or larval counts unreliable as far as the abundance of the adults in the population, is that there's asynchronous hatching. So she's laid her eggs in this site, a couple eggs there. Actually, multiple females have probably laid eggs there, and the site floods. Some of the eggs are going to hatch, but that site may dry up before the larvae can emerge as adults. And so a strategy of the species for survival is that when this breeding site floods, not all of the eggs that are in there are going to hatch during that flooding. So there may need to be a subsequent rain for more of the eggs in there that are sitting in that environment will hatch. And you know, so there may be multiple floodings to get all of the eggs that are in a container to hatch, assuming that no new eggs are being laid in that container. So it's a strategy that the mosquito has developed because they live in such a marginal habitat that's so dependent on really rain to give them places for their larvae to develop. So this asynchronous hatching with the skip oviposition means that you can't go out and count number of larvae in a container and get a reliable estimate of what's going to be flying around as far as the abundance of the population. There are some surveillance tools that we do use, though, however. There's a series of what are 80s indices. There's the house index, the Breteau index, container indices. These are all ways people have come up with to try and address the risk of dengue transmission in dengue endemic areas based on using these little jars with water in them and paper for the mosquitoes to lay their eggs on. A lot of these different indices are just a presence-absence. Sometimes they'll count the number of pupae because that actually translates better into numbers of adults in the environment. So pupil surveys have actually been shown to have some relevance of risk. Ovi traps, again, presence, absence. There are some larvae traps out there that are kind of modified black jars, and then there are adult traps out there as well. The Fay Prince trap was developed to try and catch 80s, or has been adapted to catch 80s because they are visual, and so you hang this out there and they see something and they'll go and investigate. But they're not attracted to light because they're, again, a daytime species. And then there's actually an example up here of the BG trap, which was developed specifically to try and catch adult 80s aegypti. So this is the BG trap. There are different types of Ovi traps out there. There are some newer sticky gravid traps where you put a sticky substance around the inside, so when the adult lands on it to lay her eggs, the adult is stuck to the trap, so you can kind of get an idea how many adults are in the environment. There are a lot of different traps that aren't commercially available yet or soon to be on the market, not just to catch the adult by sticking them to the trap, but also there are some traps out there that have chemicals that adult female will pick up. The chemical in the water will kill the larvae. The adult female picks it up when she skips to the next container. In the next container, she introduces that chemical into the additional containers to control the larvae there. So there's a lot of things on the horizon for 80s control, but they're not quite there yet. Surveillance for 80s indexes, this is the most widely used way of surveying for Aedes aegypti because it's so hard to catch the adults, or has been in the past. Presence-absence data is usually what most control programs that have to deal with dengue use. Pupal indices, again, pupae are more related to adult mosquitoes. You can get a more absolute measure of population density using pupae because they're only there for a day, so that habitat's probably, even if it does dry out, those pupae can still usually emerge. They'll actually speed up their pupal development if the water level is going down and the oxygen is being depleted from the water. They can sense that, and instead of taking 2 days to emerge, they can compress that down and emerge much quicker so that they don't end up dead at the bottom of a dry container. But it relies on visual search of containers with water and immatures to conduct these surveys, which means you have to go onto people's property and look at all of the trash in their yard and look for these larvae. So it's incredibly labor intensive to do these kinds of pupal surveys and larval surveys. It takes a lot of man hours. And it also requires very large sample sizes to get a reliable estimation. So how many of you have a program that can support inspecting 1,000 to 3,000 houses on a weekly basis? I'd like to see your hands. Yeah, I thought so. So while it's a very good way of measuring the abundance out there, it's a very impractical way with our resources. And this is just a little bit of data to show that the sample size that you need for number of pupae per person actually is pretty correlated, and that it does work. It's just impractical to do. So I think I'm in the to stay on time to actually maybe get you guys out here at the end of the day a little quicker. I'm going to kind of gloss over these next slides, and they have to do with surveillance. So I think Dr. Florin will probably cover maybe a little bit of this as well. But this is just more information on how pupal indices actually, there's a correlation between the number of pupae per person and percentage of pupae positive houses, and so that you can actually just go in and count the number of houses where you find pupae in. So instead of inspecting every piece of trash in the yard, you go into the yard, the first container that has pupae, you mark it as a pupae positive house, as opposed to counting all of the pupae on that property. And so you can actually speed up the surveillance that way and have less resources devoted to it, less time spent per house, but you still need to visit a lot of houses. So some of the limitations on immature surveys is finding aquatic habitats by visual inspections, because you know, these mosquitoes will actually use cryptic habitats. So in that respect, they are like Quincke fasciatus in that they like to hide. Aedes aegypti will also hide, and they will use, you know, Culex Quincke fasciatus likes that dirty water, that high organic water, that, you know, yuck that washes off the road into the storm drain, the leaky septic tank water. Culex loves that, and the word on the street is that Aedes likes clean water. Well, actually, Aedes will utilize some of those same habitats. So they have been found in septic tanks. They have been found in manholes and pits and wells and mines and storm drains and sumps. And so they are not necessarily in clean, pristine water. They will also tolerate some pretty nasty water as well. Adult mosquito surveillance. OV traps, again, there's a lot of different kinds of OV traps that have been used to try and trap out adult populations. So for surveillance, we will use these jars with seed germination paper in them to collect eggs and get an idea of the presence of the species and whether it's Aedes aegypti versus Aedes albopictus. But the numbers of eggs really don't tell you anything about the abundance of the mosquitoes in an area. A lot of people have tried to adapt this concept to trap mosquitoes out by trapping them in the jar. There are some that will use this sticky material so that the mosquito flies in and gets stuck on the sticky material. There are some traps out there people have used, you know, that polymer stuff that you can put in your flower vases to keep them moist but not have water. So you put that powder in there, and when it gets wet, it absorbs all the water. So there are people who have used that in breeding habitats to try and control Aedes aegypti. Sprinkle that powder in. When it rains, it floods, and the polymer absorbs the water, so there's no free water for the mosquitoes to develop in. And then when the rain goes away, the water slowly evaporates and it becomes a powder again. That's a strategy that people have used as a control method and have evaluated, but it's not anything that's commercially available, and I don't think that it's really been tested very well. So that's a possibility. It isn't inexpensive to deploy these ovipots, and as far as man hours, but you also have to realize that that ovipot or ovi trap is competing with all of the other natural habitats out there for that mosquito to use. And so there's a lot of studies out there trying to get at how many of these do you actually have to employ in the environment to impact a population. So how many of these different kinds of sticky traps and lethal ovi traps would you have to put out to actually control the population and to overcome what's out there in the environment. BG sentinel traps, these are for catching adult mosquitoes. They are really a visual trap, so that mosquito's down in the brush. They call these mosquitoes ankle biters for a reason, is that they tend to be right on the ground, kind of bouncing along through the vegetation, and they see a big dark shape and they think, that must be an animal, maybe a human, I'll go over and investigate. And so they go over and they're visually attracted to these traps. There's a fan in here which will suck them down into the trap as they kind of hit it and move up. These traps can also be augmented with CO2, and there's a commercial lure that you can put in there as well, which is based on mostly lactic acid, but it's trying to mimic stinky humans to make it more attracted to particularly the Aedes aegypti. One thing that you can do, these traps come with a white bag. You can actually enhance your trap catches by wrapping that white bag with black. So you make it a much better target hunting during the day. Mosquitoes don't see in color, they see black and white, so a big black shape to them is a potential meal. And so just by adding this little bit of black wrapping, you can increase your trap catches. It's useful because this trap actually samples adult abundance and catches adults as opposed to looking at larvae. It does require batteries to run the fan in there. So just like a CDC trap, you have that maintenance of batteries associated with it. And again, the putting that black wrap around it does enhance the catches for the traps. The sticky gravid traps that are out there, they target ovipositioning females. So there's a lot of debate on whether to test mosquitoes for chikungunya because you're going to see the human cases of it, and you're going to see those human cases first. The value of trapping and testing mosquitoes is more on the research side of looking at vector abundance, looking at what's going on in an area, because we really don't know, combining infected mosquitoes with feeding on other animals and things like that. So it's more of a research tool than a surveillance tool. They're inexpensive and easy to deploy, and because it's targeting these ovipositioning females, they're the ones that are going to be more likely to be infected if you are doing some sort of project that requires you to do mosquito testing. They have similar limitations and value as OV traps, but you can catch those mosquitoes out of them. So one of the control strategies is to try and trap the mosquitoes out of an area. So on top of that, you can also pull the mosquitoes out of the trap to do other things. And depending on the model of the trap, they may require more or less labor to use them. This autocidal gravid trap, AGO, was developed in Puerto Rico. Basically, it's a bucket with a paint can in it. The mosquitoes come in to oviposit, and they get caught on sticky surfaces as they go down to try and lay their eggs. So what do you do with vector surveillance data? Again, you're going to monitor spatial and temporal population dynamics and try and model that with disease transmission and disease incidence. We don't really have a good vector index for Aedes, unlike West Nile virus and Culex pipiens. The vector index kind of breaks down because it's really, really hard to get the abundance data on this mosquito because they are so hard to catch. But you can establish the habitats of the mosquitoes and their spatial and temporal dynamics, so you can use these trap types to figure out when is the actual peak biting time for these mosquitoes in your particular jurisdiction or for this time of year, or when is the population at its highest throughout the year, what months are at highest risk for large populations. These kinds of questions still need to be answered, and they still need to be answered for North Texas. Because again, a lot of the data is either very, very old or is generated in dengue endemic areas. So you can also look at relationships between weather and populations, and you can tie all of this back to disease once the disease starts being transmitted in the U.S. And then it can also help you to understand your impact of your vector control on the populations. So I think I'm going to skip over that. This is just an example from Puerto Rico where they were using these autocidal gravid ovitraps and looking at the average number of female mosquitoes in the traps per day in an area where they went in and did intensive control, vector control, IPM, versus a control area. And the bigger the circle, basically the more mosquitoes from that site, and I mean, just visually you can tell that where they were doing these intensive vector control projects, they were able to really bring the numbers down in these traps in that control area. So new tools on the horizon for the U.S. for controlling Aedes aegypti, using chlorine bleach and clay mix. That's something that's being looked at. There's a number of autocidal OB traps out there that are being looked at to be commercialized. I think there's a couple that are going to be on the market soon. Larvicides, again, perfoxafen, spinosad, how do they work for larval control? They're not the only larvicides out there. They're just being looked at for uses for Aedes aegypti. Adults, lethal OB traps, curtains to keep them out of houses, covering water storage containers. Those are messages that really should be getting out there. In a lot of ways, public health has been taking steps backwards instead of forwards when it comes to vectors. We got rid of our malaria, we got rid of our dengue outbreaks, our vector-borne outbreaks. Really in the 50s and 60s, when communities went from relying on cisterns and people collecting water for their own needs to going on to public water and public sanitation. We got rid of all of that, which was the breeding habitat for these mosquitoes back in the day. We're now, with all of our water conservation and people having rain barrels in their yards again, becoming very, very popular, they're putting habitat for these mosquitoes back into their yards in large numbers. And so educating them to make sure those rain barrels are mosquito-proof is going to be key and something that we really need to be messaging to the public. Sticky traps, there's a number of them out there. There is a project slated, if the FDA approves it and permits the company to do sterile mail releases. It's slated to happen in Key West. I don't know when, but the idea is that you release sterile males. They mate with the local females. She'll lay eggs. The eggs will hatch, but the gene that is transferred to those larvae turns on a mechanism that will kill the larvae before they become adults. And so it's kind of like the screw worm project where they still irradiate the males to make them sterile and mass release them. This is the same concept, using genetically modified males to release in the environment. And then there's also a lot of work using Wolbachia, which is a symbiotic parasite of the mosquitoes. And if you get two different strains of the Wolbachia, basically what it's doing is the same thing as the sterile male GMO mosquitoes, in that you're not getting offspring that are viable from matings. And so driving the Wolbachia infections into the population is something that is being studied, but again, it's not a commercial tool yet. So there's hope that we'll have better